Welcome back, students. Let's continue with the second part of Journey to DNA. In the previous sections, we had come across the work of Mischer, who discovered DNA when he was trying to work on proteins, and later on we found out, and he called it as nuclein. He was not aware what was that, but he saw that it was rich of phosphorus. And later on, by the studies of Levin, he did the groundwork and he found out what are the constituents of DNA, and he also gave the hypothesis of a tetranucleotide occurrence of DNA. The nuclein molecules of Mischer were being studied by Levin, and it was being explained by him, the structure of the DNA. And he also studied proteins, and he saw that proteins could be probably a good candidate to be as the genetic material, but because of their main diversity of the amino acids which could be contributed upon different proteins. But this research was still there, and later on the work of various other scientists were complementing to our knowledge about the DNA which we know today. So let's go ahead through this journey of DNA. Journey of DNA. After the work of, as we mentioned, Levin, two important scientists are well worth to be discussed here. In the 1920s, there was Frederick Griffiths. He showed that a harmless strain of a bacteria can become infectious when it was mixed with a virulent strain of a bacteria that had been killed. And the dead bacteria apparently provided some chemical that could transform the harmless bacteria to an infectious one. And this so-called transforming principle, though it was not known that day, it appeared to be a gene. Later on, Oswald Avery, as well as his colleagues of the Rockefeller Institute, took over the work of Griffith, and in 1940s they found that a pure extract of the transforming principle was unaffected by the treatment of proteases, RNases, and they found that it was affected by DNases. And they identified that this molecule, which was getting transformed into the which was converting the bacteria to a virulent strain, was being found to be as DNA. And they identified DNA is the genetic material rather than protein. So let's go into the details of these experiments of Griffith as well as Avery and his colleagues. Let me introduce you to Frederick Griffith. He's a British bacteriologist who was searching for a vaccine against pneumonia during the post-Spanish flu time. It's quite interesting today when I talk, because right now we are facing a pandemic, the COVID-19, and about 100 years back there was a Spanish flu in 1918 to 1920. And after the pandemic was over, by around 1928, Griffith reported the Griffith's experiments. Now what was he doing? Okay, he found that some principle or some information was being transferred from one bacteria to the another, converting it into an infectious agent, as we mentioned earlier. And this process was known as transformation. So let's go into the details of that. So let's go into the details of this using an animation provided by the DNA Learning Center, the Cold Spring Harbor Laboratory, with its pool of animated videos which are available online for the reference of students. Now this is Oswald Avery. His colleagues did a series of experiments using the strains of pneumococcus bacteria which causes pneumonia. Pneumococcus grows in the body of the host, but like other types of bacteria, they can also grow on the solid or as well as in liquid cultures. Prior to Avery's and his colleagues' work, in 1928, we know that Frederick Griffith published a study on the different strains of pneumococcus. Two in particular, the S and the R, looked different. The S colonies had a smooth surface and the R colonies looked rough. And the smooth colonies looked smooth because that each bacteria had a capsule-like coat made up of sugars. And this coat protected the S bacteria from the host immune system. And so the S strain is being found to be infectious. Whereas the coatless R strain is not been found to be infectious. And Griffith found that the mice injected with the S strain could develop pneumonia and die within days. So that's what he is injecting the mice with the S strain, and he's seeing that within 3 days probably the mice were being killed. Now the mice injected with the R strain or the rough strain do not get pneumonia. And Griffith also noticed that different strains of pneumococcus could be cultured from one patient. That is, and he began to wonder if one strain could change into another, and to test this data, he did a series of experiments using the R and the smooth strains. So first what he did is he heated the S strain culture to kill the bacteria, and as he predicted, when it was being injected into the mice, the heat killed bacteria did not cause an infection. The mice was running. So when he killed the S strain, it was not causing an infection. So Griffith co-injected the heat-killed S with a live R into the mice. And much to his surprise, the mice developed pneumonia and it died. Even more astonishing, Griffith was able to isolate live S strains from the blood of the infected mice. So he's finding that though he had killed the S strains when they were injected into the mice, in the blood of the mice there was being found to be live S strains when they were being inoculated with the R strains. And these cultures could infect other mice too. And the S strain cultured from the infected mice remained active, showing that the change was stable and it was inherited. So that's it. Though you had killed it, it was continuing to be as though the S strains were being first killed and then they were being inoculated with some R strains. It has been found that they were being converted into the S strains further on, and this was remaining active and showing that the change was stable and inherited. So Griffith concluded that some principle was transformed from the heat-killed S to the R strain, and the principle transformed the R into the infective S strain with a smooth coat. So this is a rough strain that you have, and some principle was transforming this rough bacteria into a smooth one. And what transferred it? That was a question during that time. Now, Avery and his colleagues, mainly Mc Loed and Carthy, they began experimenting using this test tube assay instead of mice. They used a test tube assay. Okay, so they took the smooth strain which were being killed, okay, and they used detergent to lyse the heat-killed S cells. And what happened is, further on, they got the lysate, and they took the lysate and they inoculated into a separate test tube. And what they did is they mixed the lysate with the S lysate with the rough strain. And to the astonishment, the test tubes assays worked well, and they showed that the heat-killed S lysate could change the R to the S, and the transforming principle was something in the lysate. So they tested each of the lysate components for the transforming activity. First, they incubated the heat-killed S lysate with an enzyme called S3 which was something which would chew up the entire sugar coat. And so the enzyme S3 was being, so what happens, and the mouse, you can see what are the contents in this tube. So all the coat samples were being gone when they put S3, and further on, what's it, no coat was there, and so they did, the coatless lysates were taken and they were being further mixed with the rough isolates. And they found that even now when the coat was being killed, it was being converted into a what? S strain. So the coat did not contain the transforming material. So then they included the coatless S extract with a protein digesting enzyme, trypsin and chymotrypsin. And that was further mixed with the rough strains. Okay. And they tried to test the lysate's ability to transform. Here also, now you can look into the tube, the coats are being removed, the proteins are being removed here, and but even now they can see that even if the proteases were being acted, something was transforming the rough strain into a smooth strain. So then they precipitated the nucleic acids DNA and RNA with alcohol, and they were the first to isolate nucleic acids from pneumococcus, Avery, Mc Loed and McCarthy. And now they took the alcohol and they got the DNA and RNA, the nucleic acids were being obtained. And what they did is, the transforming principle was not sugar coat and the protein. They suspected maybe one of the nucleic acids were responsible for that. So they dissolved the precipitate of nucleic acids in water and tested the transforming ability of the solution. So they had immersed it and they took some amount of the nucleic acids, and RNA was destroyed by using RNase enzyme. So the coating, no coat was there, no proteins were there, and no RNA was there. And again they took a part of this and they tried and mixed it with the rough strains, and even then it was being converted to the smooth strain. So what was the thing left over? Then they identified probably it could be pure DNA, and the final test was being done by using a what? DNA digesting enzyme or a DNase. So in this tube there was no coat, no proteins, no RNA or no DNA. And what happened as a result of it? When this was being taken and mixed with the rough strain, they came across that the mouse, when we clicked, so you can see it still remained as a rough strain, that is, it was not being converted into the rough strain, into the smooth strain, sorry. So Avery, Mc Loed and McCarthy together conducted that DNA is the transforming principle, and they published their result in 1944. I hope this session is clear to you that right now we identified that something was transformed, and the transforming principle was identified to be as DNA by the experiments which was being done by Avery, Mc Loed and McCarthy. Thank you for the session, and we will continue our journey in the coming sessions. Thank you.